name is Mark Glover. I'm a professor in the Department of Biochemistry at the University of Alberta. I want to talk to you today about some of the research going on in our lab where we're trying to understand how genes are expressed within bacterial cells. So as we know, bacteria, of course, have DNA like all cells do. And the DNA is regulated in different ways in different kinds of cells, but we are studying a particular kind of process that works in bacteria, and that's very important for many different kinds of functions that bacteria carry out. Now, our research is really emphasizing is the role of a particular kind of protein in the control of this gene expression. These proteins are called RNA chaperones. So how does this control work? So it starts with the DNA, which of course is the molecule that makes up our genes. So here's the familiar double helix. The way genetic information is expressed is through a series of processes that start with something called transcription that involves a protein complex called RNA polymerase. So it's a set of proteins that form this sort of complex that bind to the DNA and then run down the DNA, and in doing so, they make a new molecule called messenger RNA. So this is another form of nucleic acid, although it's single-stranded, not double-stranded. And so here's a sort of cartoon diagram of what that looks like. So it's got this single-stranded region. At one end, it's got something called a transcription terminator. So this is where the transcription normally ends, and it's got this little bit of double helix and this sort of hairpin structure. So the RNA kind of bends back on itself and forms this little bit of duplex. And then there's a little single-stranded tail at the very end. So this sort of terminates a lot of these mRNAs that we find in bacteria. So this is a mediator of information in the genetic process. And the next step is something called translation that involves another protein complex called the ribosome. So the ribosome binds to the opposite end of the RNA from this terminator structure, and it runs down the RNA, and in doing so creates another chemical chain called protein. And so the proteins, of course, we're all familiar with these. These are actually the functional units within cells, and they make up a large proportion of cells, and they carry out a lot of the functions. So this is the translation process. So ribosomes interacting with the messenger RNA to translate and make protein. So this is the basic process in bacteria, and in fact, it's very similar in eukaryotic cells as well. But what we've come to understand is that this process is regulated by another kind of RNA molecule, which we've called small RNA or sRNA. So that's what these look like. So sRNAs, they're smaller than the mRNAs. They're a lot shorter, but they have the same overall structure. They terminate with these transcriptional terminators, things with these hairpins and the little single-stranded tail on the end. And what our research has been focused on over the last several years has been understanding how these interactions work to regulate gene expression, and moreover, the role of a family of proteins that enable this recognition of the mRNA by the sRNA to actually happen. So these proteins are called RNA chaperones, and there's a particular family that we've been looking at of RNA chaperones called the FINO family of RNA chaperones. And some of our most recent work has revealed part of the role of what these FINO proteins do. And what they do is they seem to actually bind and block the translation by binding to this transcriptional terminator on the sRNA. So here's the FINO protein binding to that transcriptional terminator to block translation. And in our most recent studies, what we've been doing is trying to visualize precisely how this works. So we've been using experiments to get at the precise three-dimensional structure of FINO proteins bound to these sRNA terminator structures. So the kind of experiment that we use to do this is called X-ray crystallography. So it's a way of imaging at atomic resolution the structures of molecules. And so it's kind of like microscopy, except it's different. So microscopy uses regular light, which is actually, the wavelength of light is too large to actually visualize molecules. So we have to use radiation of shorter wavelength, and the shortest wavelength we have is X-rays. So we've been using this method, X-ray crystallography, which gives you the atomic resolution because of the short wavelength nature of the radiation. Now, there's some tricks in this method, and one of the biggest sort of problems or tricks is that you can't just image the molecules directly. You have to actually make crystals out of them to get enough scattering power to actually visualize what's going on. And so we've been able to do this. So we've been able to crystallize one of these phenoproteins bound to one of these RNAs. And so here's one of these crystals here. These things are pretty small. They're only a few hundred microns in size, but big enough to do the experiment. And so we mount these crystals in an experimental apparatus, sort of like this, where we have the X-rays coming out, and then we mount the crystal, the X-ray beam hits the crystal, and then we get scattering. And what we end up with the scattering is a pattern of spots that's shown here. So this is actually some of the scattering from one of our phenO-RNA crystals. And of course, this doesn't look anything like a real structure, because really what this is the unfocused image of the molecule in question. And so we have to do some computational processing of this kind of data in order to regenerate the real three-dimensional structure in the computer. But we've been able to do this, and we've been able to get a structure of one of these phenoproteins bound to the RNA target. And so I've got a movie in the next slide that shows you what this looks like. So here's the structure of the phenoprotein. The blue is the positively charged surfaces on the protein, red is negative. This is particularly important because the protein largely interacts with its RNA through electrostatic interactions between the positively charged surfaces on the protein and the negatively charged RNA. It's also going to use this little sort of depression here on the surface. So here's the RNA coming in. You can see that 3' single-stranded tail of the RNA docking right into that pocket. You can see all this positively charged surface that's relatively close to the RNA hairpin structure here. So this is starting to show us a picture of how this protein can specifically interact with this structure that occurs at one end of these RNA transcripts to bring the protein into proximity. So these experiments have showed us how these kind of proteins can bind and recognize these transcriptional terminators in these RNA structures. But there's still unanswered questions here. And the biggest unanswered question is really, how does the protein facilitate the interactions between the small RNA, the sRNA, and the messenger RNA? So we know how they get there, but how do they facilitate the RNA-RNA interactions? And what we think is going on here is that there's something called RNA structure remodeling going on. So RNA is kind of a structurally plastic kind of molecule that can adopt lots of different shapes. And we think that the sRNA and the mRNA have to get into the precisely right kind of conformations in order for them to interact. And we think maybe this is what the phenoprotein is doing. So the interaction that we visualized is showing us how the phenoprotein gets there in the first place. But then it's got to be doing something else to allow the two RNAs to actually associate with one another. So that will be our ongoing work. And to finish, I just wanted to mention the people in my lab who did this work at the University of Alberta. This is a nice picture of some of the ice fields on the western edge of our province in the Rocky Mountains in Canada. And I also wanted to mention some of our collaborators. So collaborators at the University of Innsbruck, Martin Tollinger and Christophe Kreutz, as well as a group at the University of Lyon, headed by Xavier Charpentier, and our funding agencies.